Hello and a very warm welcome to Cognito Key. Today we are going to unlock the secrets of a truly incredible technique. It's like a super precise sorting machine for the very building blocks of life. It's a cornerstone of modern biology, and it's called electrophoresis.
So just imagine this for a moment. You have this microscopic soup, a mix of DNA, proteins, all these things that are so incredibly tiny, they make a single grain of sand look like a mountain. So the question is, how on earth do you sort them out? How do you see what you've actually got? Well, it sounds almost impossible, right? But science, as always, has a very elegant answer.
And that solution is a process we call electrophoresis. The simplest way to think of it is as a high-tech filter for the molecular world. It's a tool that lets us sort, identify, and actually see the very stuff that we are all made of.
Okay, so how does this amazing molecular sorting machine actually work? Well, it's powered by a force that all of us are familiar with, electricity. An electric field is basically the engine that drives this entire process, giving everything the push it needs to get moving.
You see, it all comes down to a very fundamental rule of physics. Opposites attract. In an electric field, any molecule that has a negative charge is going to be drawn towards the positive pole. And any with a positive charge, well, they're going to head straight for the negative pole. It's this simple push and pull that gets the entire sorting process started.
But, just moving molecules from one side to the other isn't very useful for sorting, is it? To separate them, we have to introduce an obstacle course. And this comes in the form of a gel, a special material filled with a network of microscopic pores that acts as our molecular filter.
And this is where it gets really fun. It's like a molecular race. The electric field is the starting pistol, and that gel is the racetrack. The smaller, more nimble molecules, they just weave through the gel's pores so easily, speeding right ahead. But the larger, bulkier molecules, they get tangled up in the traffic and move much, much more slowly. So by the end, they're all lined up, perfectly sorted by size. It's brilliant.
Now, this powerful idea actually has a pretty long history. The basic principle was first noticed way back in the early 1800s, but the real game changer came in the 1950s with the introduction of that gel matrix we just talked about. That single innovation is what turned a simple observation into the precision tool that labs all over the world rely on today.
So naturally, over the years, scientists have adapted this core idea for different jobs. While the principle of sorting by charge and size is always the same, the medium can change, from simple paper to incredibly thin capillary tubes. But by far the most widely used and versatile method is gel electrophoresis, and that's what we'll focus on.
Gel electrophoresis is really the undisputed champion in labs everywhere. It's the go-to method for scientists who need a clear, powerful, and really reliable way to separate and analyze the molecules they're studying.
And of course, not all gels are created equal. You have to pick the right racetrack for the right runners. For sorting big, long molecules like DNA, scientists use a gel with a looser weave called agarose. But for smaller things like proteins, they need a much tighter gel in a technique called SDS-PAGE. This ensures the proteins are sorted purely by their size.
So, let's just walk through a classic example, separating DNA. First, the DNA samples are carefully placed into little pockets in the gel called wells. Then, you flip the switch and the power comes on. Now, because DNA is naturally negatively charged, it immediately starts its journey toward the positive end of the gel. When the race is over, a special dye is added that makes the DNA glow under UV light, revealing this clear pattern of bands, all sorted perfectly.
So, why does all of this lab stuff matter so much in our daily lives? Well, in forensics, it's what lets investigators create a unique DNA profile from a tiny sample to match a suspect. In medicine, it helps doctors spot the protein markers of diseases, which leads to faster diagnoses. It's also absolutely crucial for making sure the medicines we take are pure, and even helps keep our food safe by detecting contaminants.
Ultimately, what electrophoresis does is something truly remarkable. It makes the invisible world visible. It lets us read the molecular story of life itself, from a single gene to a critical protein, and then lets us use that knowledge to build a healthier and safer world for everyone.
So, we now have this incredible power to sort and understand the very code of life. And this just opens up a whole universe of possibilities, leaving us with a really exciting question. What great mysteries will we choose to unravel next with this amazing tool?
If this explainer helped you unlock a new piece of the scientific puzzle, please do show your support by liking this video, sharing it, and subscribing to Cognito Key. We are really dedicated to making complex knowledge accessible to everyone. Thank you so much for joining us.